I was today. I was reading the book of Wilson and Walker, and I saw there's so many techniques are left for you, like spectrophotometry, X-ray crystallography, uh, NMR. But I will not. I think I will put them in the proteomics lecture. So I'm also taking a proteomics section of pure proteomics, where I will be teaching you various proteomics techniques, which is quite different from the molecular biology techniques. So I want to save them for them, actually, for those students exclusively. So I might not share here, but there are some chip sequencing, chromatin immunoprecipitation, and ELISA is very common. Then immunoprecipitation, some more other techniques were there that I didn't share. Anyhow, let's start our lectures today then with this. So all good students, you are all with me now? Yeah, okay, let's start the session then. So mainly today we are done from our first day. We started with the basics of molecular biology. Then we slowly and slowly went through the basics molecular biology techniques. Then we came to the recombinant DNA technology techniques. Now today, after done with the DNA and RNA and all those NGS and many other aspects of MBBT and biochemistry technique, now it's come part of proteins. Sorry. So to check what happened at the protein level with the protein with the isolations, this quantifications, and then purification. So there should be also quantification should be also be there. Mhm. Why it's not coming in the middle like this? And this should go there. Yeah, so these three different parts will be dealing with you. And then we deal with the uh, homogenization, how you homogenize your samples, then how you fractionate it out of it, then centrifugation, then various techniques about quantifications, and then how to purify it with the help of chromatography, electrophoresis, also a little bit about mass spectrometry, MALDI-TOF, and that's it. That's all I will be dealing today for today's lecture, and I keep it quite interactive and nice. Oh, sorry. Okay, let's continue then. CRISPR-Cas9 is left, so I will do it maybe later. One video about that. So, um, so these are there are various famous Nobel Prize winners have been there in the field of chromatography and electrophoresis. So few of them I feel like to share their names and what the work they did in chronological order. So you can see here Porter Martin, Archer John Porter Martin from United Kingdom. They invent the partition chromatography initially. Then comes our Millington Singh at the Scotland Institute in the Robert Research Institute, Bucksburn in Scotland. They also invented this partition chromatography. So they both shared together the Nobel Prize winner for the same. And then comes the Arne Willem Corin Tisselus, 1948, from the Uppsala University. So he worked on the electrophoresis adoption analysis, especially for the discovery concerns in the complex nature of serum proteins. Then in 2002, recently, John B. F. Fenn, he found the development of identification of and structure analysis of biological macromolecules. So he developed a method for the same. So these people are known for their mass spectrometry and sedimentation techniques. Tanaka from Japan, he developed a ionization method for mass spectrometry analysis for various biological macromolecules. So he also shared his Nobel Prize winner with other concerned persons. So Japanese, if you see from the research point of view, just want to share one human trait. They are really strict about the studies what they're doing. If their paper is retracted means something is wrong, and the agency says that whatever you have done is fraud, and it's not good enough. They tend to kill themselves on the same day. They're just known famous. Japanese are famous for their self-respect. Once their self-respect is lost, then they are like they are not want to live anymore, and that's how they're committed to your work. So that's how the Japanese work culture is really respectful to each other, and what they're doing with full of honesty. So further, Theodor Svedberg from Uppsala University, he also found work on the disperse system in 1971. Then in organelle fractionation, these are famous scientists like Albert Claude, Christian de Duve, and George P. Plade. So they discovered in the each year some to check the structural and functional organization of the cells. So they got the each year their Nobel Prize winner. Then Sanger we know for his work on the proteins, especially that of insulin, Frederick Sanger. And then other is also in 1980. He's known for contribution to the base sequences in nucleic acids. So they also got Nobel Prize winner for the same. It's good actually when you're studying, when you got a chance to work with a big scientist or Nobel Prize winner. When I was working, we were able to see a Nobel Prize winner from Göttingen itself. It was in 2016, I guess. I forgot the name. His work was something with the ion optics, with the microscope. He discovered a really good technique in the microscope at that time, and three people shared the Nobel Prize winner. One from USA, one from UK, and one from Germany. So these three people together, they worked very hard. And once you get the Nobel Prize winner, your doors are open to even more research, to do even more things that were limited to you, because funding is a big problem in research. But once being a Nobel Prize winner, then you can widen up your research further. Then this is to start with something with the protein assay. It's written, it's a biochemical or biological means used to monitor the presence of targeted proteins, to check how much it's present, to check that specifically. For example, enzyme activity, specific labels, functional properties such as binding, they are all present on that. So like let's say lactate, we add NAD plus, and then lactate dehydrogenase as a enzyme. So what it will do, it will change from lactate to pyruvate, and then NADH and NH plus. Same way, EL5A precursors are being added with the deoxy or uh, deoxyribozyme with the hydroxylase. So both have different reactions. In first case, we have produced deoxybutyl ELF alpha, then other is hyposyl ELF A. Then comes our, that was something to show you in a little bit about protein assay. Then comes the protein isolation. So there are two different steps to do so. First is homogenization, that is keeping your cells in equal amount, that you disrupt your cells. Disruption is there, distortion is there, and there's a homogenization, like everything is quite similar within the append. Then fractionation comes, in which your crude separation of homogenates are separated into various fractions, and that's known as fractionations. So here in the homogenization, there are like four way of doing it. One is Downs homogenizer. It's a technique where you can homogenize things automatically. Then is a sonication with the sound, or giving a osmotic shock it, or giving a French press it. So there are various techniques to do so. Then fractionation is possible by a pH, or by heating it, by changing the pH or by changing the heat. We can do this fractionation pH also by ammonium sulfate precipitation or differential centrifugation. Doing a different way of centrifugation, we can also have different membrane bound or vesicle bound particles into our system. So here you can see on the right hand side, you have a homogeneous forms of your cell culture. your cells are there. Then you centrifuge for 50 G for 10 minutes. So you have first of all initially a supranatant, yeah, and then at the bottom is the palette. At the first, when you do this much of homogeneous form and supranatant in that form, so you have nuclear fraction. Okay, you have a nuclear fraction. Then if you keep increasing the speed of the same sample to 10,000 G for even double the time, 20 minutes, then what you see is uh, uh, in the palette is mitochondrial fraction. Then in the palette you have all the mitochondrial fraction. Then even further you increase again 10x speed of the same 100,000 G, and even for 1 hour, then what you see uh, in the palette is your microsomal fraction. So all the microsomal organelles will be present, and the rest on the top will be your cytosol left. So that's how with the different G's you get in palettes different things, and that's how the uh, yeah, you can collect things differently. And then comes the centrifugation. This is to separate particles and macromolecules based on the mass, density, and shape, based on the three characteristics such as uh, mass, density, and shape. You can differentiate of organisms. So again, this technique here is showing you differential centrifugation. On the right is a rate zonal centrifugation. So on the left side, sample is poured on the tube. So sample, more dense particles, less dense particles are there. Then there's centrifuge, and you can see the centrifugal force on the right hand side. Particles started to set down, so it's not fully 90 degrees Celsius, but somehow tilted at 45 degrees Celsius. So because of that, your palette is there left down, so you have supranatant, you can decant into liquid container, and then you can remove the, you can have the palette, and whatever the palette according to the centrifugation speed, that will we will have it that then furthermore, in the rate zonal centrifugation, you have a gradient. First of all, like here in this case is a sucrose gradient. Okay, there's a sucrose gradient. In the sucrose gradient, we added our large particles and small particles, a mixture of large and small particles, and we want to differentiate between them. And then we centrifugate, and then particles got settled according to their mass. So the heavy one will be down, and the small one will be here, like that one. Then you stop the centrifugage, and then you can see you can collect these samples into different tubes by pouring a hole downwards and collect fractionation to do your assay. So your samples will be increasing mass of particles like this, keep going out. So that's the main difference. Further, these are the various speeds I have. It's written like at 300, 3000 G, 10,000 G, 40,000 G, 200,000 G, and 400,000 G. So what for how long? If you run a sample for 24 hours, it will be protein of around 50,000 Dalton. If you run on this speed, then you will have ribosomes, PDH, proteins complexes of less than 500,000 Dalton. If you do it for 30 minutes in this speeds, then you have membrane, plasma membrane, microsomes, peroxisomes, etc. On this speeds, you have mitochondria, inclusion bodies. On this, you have pallets with the large organelles. Then, while doing a centrifugation, there is a thing called Swedberg equation, Swedberg equation or sedimentation coefficient. So this is your Swedberg equation, actually this whole, and this S is your sedimentation coefficient, and it is known to be equal to the 1S equals to the 10 to the power minus 13 seconds. So sedimentation velocity technique. Now comes the third technique. First, we are done now. What does it do actually in this case also? We have low density here, yeah, and we have a high density solution. Okay, and then we take a centrifuge tube and we create a density gradient. So first we add a high density solution, then we on top we add a low density solution. Then layering of samples. So on top you add your samples, Then rotor is run like this. So there's sediment separation by sedimentation coefficient. So all your organelles will be separated according to their sedimentation coefficient, according to the density. So then afterwards, according to them, we will collect them according to their size. So we keep pouring them like this. First is uh, like we said, so first you will have low molecular mass, then we will have higher molecular mass. So we keep pouring them like that. So these are some of the protein molecules with their respective Swedberg units and their molecular weights that you can can find out in your reactions. Further, mm, yeah, how these molecules, as I said, their sediment coefficient is directly proportional to the density. So here you can see various molecules. Like microsomes, mitochondria, chloroplast are placed as per the graph. Now comes the part of once the isolation is done of a protein with the centrifugation, fractionation, uh, sedimentation coefficient, with all the previous techniques that we have discussed. Now it comes to the quantified, like how much protein do we get out of our experiments, and then we can go for further, because you have to make a control, as I always tell. You have to see control, and you also have to see uh, अपना your samples. So you both need to be equal amount, and to check and to quantify them, mostly we use Bradford assay based on the Commercy dye, where you make a sample of like five different concentration in increasing concentration as a control, and you put them in your sample and check the color first of all, how is it like, and with the help of spectrophotometric technique you can answer the that how much concentration is your sample is. To do so, let's start with the video.
In this video, you will learn how to perform protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled Sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through, and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample, and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes.
Hello class, is it okay? So I think some so far is it okay? Could you just give me thumbs up if all is fine with the class? Okay, I thought maybe. Oh, let me know from where shall I. So we were at the empty.
Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample.
So here class you can see at 1 microgram per ml is the concentration that is equivalent to the milk concentration, and others colors are not similar to that. So that's the basic principle behind it. So in the control you can see how brown color it is, but it's got quite middleish blue color. So I will continue.
To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled control. Use the blank to set the spectrophotometer to 0 absorbance, or 100% transmittance. If instructed, Read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
So that's it. All clear. I found Bradford method. I use in my PhD whole PhD Bradford method. I used in my PhD whole Bradford method. I found it pretty good, and my data was always pretty much quite good actually. So I never went to any other technique. Okay, Fallen Lowry might be more specific, but in our lab we have Bradford in huge amount. Maybe that's why we were we were using it. So sometimes it happens from lab to lab. Some lab use Fallen Lowry method. Some labs use Bradford method. So both methods could be used according to their things. Yeah, there is a formula for that. So you have to make a linear graph, a linear regression graph, and you can, with the help of your sample value, first you make a linear graph with your control samples, with the standard samples you have added. You make x versus y graph, and then you can put your samples to that, and then you can calculate the concentration with respect to y-axis. So x-axis will be concentration. Yeah, so like so far I remembered. Burette reagent. I have no idea, bache, Shivani. Burette reagent, what's that? So that's the basics of this. So let's continue. So before we continue, or I do that at the end. Then all the PyMol thingy and CRISPR-Cas9 and gel electrophoresis virtual workshop. We do not stop the flow of our workshop today. Then. So we're done with this. This is also another method of protein quantitation, that is with the help of ultra sensitive fluorescence based protein quantitation, which is done with the help of nano orange protein quantitation assay. It's done by Jones LG and Hopel RP Singer. So you can check them at the website if somebody is looking for this method. So in this method they just add dye to the samples, heat for 10 minutes, cool to the room temperature, and just reduce fluorescence. That's it. Nothing big science behind it. That's it. And then this is, I was telling you, on the bottom is your protein concentration, on the left is y-axis, the fluorescence coming out of it. And then you make a curve graph like this, linear regression, and then you can check with your sample size, whatever your fluorescence that you see for your sample, then you can see the concentration is that much. As simple as that. This graph is created with the help of standard curve. So now we are done with the isolation part. We are done with the quantitation part, quantification part. Now comes the purification part. So we have a lot of different way of purification of your protein, via chromatography, via dialysis, via HPLC, via mass spectrometry. So there are various gel electrophoresis, various methods are there. So we will discuss them one by one today. So in this dialysis method, it's work like simple as dialysis. You have a dialysis bag in which you have a concentrated solution. Outside is buffer solution. So inside that solution, you have a big proteins and small proteins, and you want mostly big proteins to be get out from your sample. So to do so, they make equilibrium first, but at the equilibrium, most of the smaller particles will go out, and inside you will have mainly the big protein particles, and you can extract them actually. So to this is also Proteo Plus kit, which is used in the market, sample for the matrix assisted laser desorption and mass spectrometry, MALDI-MS it is used for. So before going to MALDI-MS, you put your samples, your dialysis is happening within the tube here itself. So your bigger particle proteins remain there and the smaller will remove like that. So this is also something about dialysis, a short video about it, how it is done in the laboratory. So that was very simple and easy method to check via dialysis you can extract your pure protein. Then comes the chromatography. So liquid chromatography or chromatography, they are used to differentiate between your big protein and small protein on the basis of charge or size or ion exchange. So all these different ways of exchanging the between big particle and small particle. So liquid chromatography is a term which refers to all chromatographic methods in which mobile phase is liquid. So mobile phase is the liquid in that case, and stationary phase may be liquid or solid in that case. So first one is gel filtration electrochromatography. So in this, what you do is first of all you have a protein sample up. Uh, then there is a molecular exclusion gels down there. So you have a gel system here downstairs. So what you can see in these different steps, when you add different proteins, so you will have carbohydrate polymer beads. Yeah, these are carbohydrate polymer beads, and these are small molecules to enter the aqueous spaces within the beads. These are small molecules which enter into the beads. Yeah, and there are large molecules which cannot enter to the beads. So the one which cannot enter to the beads, they will flow down like that. The one with the beads, they will attach to it and will not go further. So same way you can see we have a layer large to explain the same thing in another way. You have large proteins and small proteins on top, layer of samples on column. Then you add a wash buffer to remove the through the column. Then you can collect fractionation. So you can collect at different level all these different proteins. At first stage you will have liquid. At second stage you have big molecules. At the third stage you have the green molecules. So like this. So in the while doing so, the gel that we use here is the Cefadex. This is the bigger gel G75, and this is small one G10 with less kilodalton of size. And you can see this blue color of your product that you're looking for being differentiated slowly. First first the gel is removed, then slowly in the second beaker we can get our samples. So various gels that are available for gel filtration chromatography: Cefacel, Cefadex, Toyopearl. So each have different exclusion limit of size. So it's like 4, 40, 100,000, 100,000, 200,000, 5 lakh, like this way. Each has different polymer base: agarose, cellulose, dextrin, ethylene glycol, methylene. So this is to explain you now a small video.
We're starting with gel filtration medium. This is Cefidex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins above 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the speaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now I'm going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of 2 molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1 million. Fluorescein, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these 2 molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescein, 400 microliters of the blue dextran. Nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation has proceeded somewhat. I'm going to try to add a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50, and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. Still got a little overlap. So I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the dextran that's in the column if you're storing it over a long period of time.
That's it. That's why I thought students. Mhm. This one. Because I try to explain each thing with animations first, then one video is necessary because having a virtual lab here, it's quite because things are quite understand to see on the 2D thing, but the way we understand these FedEx G50 like straightforwardly, that's how the thing is happen. So these are really informative and very nice things to go for. So we keep the same trend and continue. Um yeah, now ion exchange chromatography. So in this, बच्चों, what happen is is that there is a anion or cation. Either it could be cation exchanger chromatography or anion exchanger chromatography. In this one, if your bead positively charged protein bonds to the negatively charged beads. If your beads are negatively charged, then it's a cation exchanger. If your beads are positive charged, then it's an anion exchanger chromatography. So main principle in all the chromatography that you have studied is based on their size. Yeah, ones who are touched to the beads, or the one with the bigger size, they can go later. One with the smaller size, they go faster, and then you can differentiate according to the color. In this case that we have seen, in this video, what we have seen is according to the color, or in this case according to their charge. So whatever the protein charge is, whether it's a protein charged or a protein is positively charged or negatively charged, they will come to the they will come according to that. So protein in a solution has a low ionic strength is added to the column. The protein interacts with the beads via electrostatic interaction. Then resins carrying positive charge are also called an ion exchanger, because an ion will exchange in these type of columns, and the resins carrying negative charge are called cation exchanger, because cations exchange on this type of columns. So you understand the main difference between cation and an exchange. So in this case, you can see the positive charged ones got attached to the negative, whereas the negative ones, they didn't attach to it, they mostly flow with it. Then later on, with the help of elution buffer, we remove them and get our targeted proteins out of it. So same thing being repeated. There is a carboxymethyl group. There is a ethyl methyl ion group. Both could be used for it. And in an ion exchange and an exchange chromatography, matrix is accordingly positive and negative respectively. Further, in the ion exchange chromatography, sulfonate S and quaternary amines, diethyl aminoethyl hydroxy, alpitite. So these are various groups are used in ion exchange chromatography for the various exchange type of such as strong cation, strong anion, weak anion, and strong anion respectively, and the structures are as respective. So one video for that also. Then we take 5 minutes break.
The separation of ions and polar molecules based on their charge. As well as other forms of chromatography, in ion chromatography, there is a mobile phase, which generally consists of buffer solution. And there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into an anion exchange chromatography and cation exchange chromatography. In anion exchange chromatography, negatively charged molecules are attracted to a positively charged solid support, which is usually beads that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. When the column is equilibrated, the second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged. And the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge. And at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge. And both protein B and C carry a net positive charge. In addition, protein C is more positively charged, since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography, protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column, as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained, and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also, varying the pH of the buffer can be used to affect the separation, and to elute the retained proteins. As we seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B, and below the isoelectric point of protein C, the protein B will become negatively charged, and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified, concentrated form.
Okay. So that was thing with the cation and anionic exchange chromatography with respect to pH point of view, which we didn't discuss it before anywhere. So the elution here was done with specific to the pH. According to pH, they were like getting out with the isoelectric point, as we can see with the pH 8, 3, and 7. So that's how it is done. So now we are done with the We are done with gel filtration chromatography. Then we're done with ion exchange chromatography. And now comes the affinity chromatography. This method mainly relies on the knowledge of properties of the enzymes or proteins that you want to isolate. Either antibodies must be available for that enzyme against that enzyme, or enzyme must be known to bind to the some substrate such as ligands or non-covalently in the reversible way. So it is the most specific chromatographic method so far because specific binding is done to the target proteins with the specific ligands. So here is one cartoon of the same. So you can see glucose binding protein attached to the glucose residues on the beads. There they are attaching. Then addition of glucose, we add additional glucose to it. Then those glucose binding proteins are released, and addition of glucose as we added that, so they got attached to the beads. Then later on we can remove them according to the elution factor. So again, in this affinity chromatography we loaded pH 7 in buffer, a protein recognized by the antibody. These are the proteins which are recognized by the antibody, and the blue are the ones which are not recognized by the antibody. So then we wash them and then elute them with the pH buffer 3 later on. So again in the same principle. So most of these chromatography principle is same, quite not so much different. Some we use an ion exchange, a cation exchange, but in this case also according to the pH buffer, they could be ruled out of the things. So there's one drawback of this chromatography technique, that it can often be so strong that very hard conditions are needed to elute the enzyme. So that's one of the thing. So this lead to the denaturation of the protein, okay, which is not good for our experiments. Then non-specific bindings may also occur, especially where large ligands such as antibodies are employed. Therefore the non-specific bindings can overwhelm the correct binding sites at very high proteins, leading to the poor purification. So therefore this is often limit to the protein concentration that can be applied to affinity column. So one of some
Depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and elution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
That's it. Very small video. So just let's have 5 minutes break now. So I see you soon with HPLC part. Just get refreshed up. So it's 1913. I see you at 1917. So, class, I'm back with the next session of HPLC. After done with the so many techniques of liquid chromatography, various different types, now it comes the most advanced version of chromatography where we had high pressure liquid chromatography where we would, with the help of pumps, a high pressure for our samples, and instead of having a solid phase, they go through the capillary C18 capillary tubes, and from there the separations tend to happen of our samples. So in this case, high pressure liquid chromatography is the most enhanced column method for protein separation. Resin signs are more refined, like C18 size of used in this case. All the chromatographic methods described above can be applied with HPLC column and equipment, but with HPLC separation peaks is almost always better than the standard chromatography column. So the results that we see, the resolution that we see with HPLC is almost the best one. An intermediate version is there is also between HPLC and other column method is called FPLC, that is fast performance liquid chromatography. So that is not so important, but you should know. But you should know. Okay, these are also available in the market. So in HPLC, the column is way too large, containing small particles known as stationary phase. Chromatic bed is composed by the gel beads along with inside the column. Sample is introduced into the injector, then carried into the column by the following solvents. Once in the column sample mixture is separated, as a result, different columns adhere to the diffusion into the gel, as I explained before, and these zones are referred to as bands. So as the different materials are separated, so we see them in the results at the different absorbance with the time, these bands refers to the zone levels, and we can tell about like how much our sample has been eluted. So little bit about HPLC now.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography.
Let me just
It is
increase
used
the
for
size of the video. Whenever you are working with any figure, always work from the right corner bottom.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase nonpolar. In paper chromatography, the solvent moves along the paper by capillary reaction. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time.
So here you can see in the HPLC you have peaks coming up, and the one is the more polar will be later, the less polar is first, and then how your peaks are being generated. So this is how you can separate molecules.
1.5 minutes represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
Thank you very much. So thank you, students. So So that's it for the HPLC point of view. So this is another method of fast, as I discussed, this is something in between column and HPLC. This is fast performance liquid chromatography, which is a type of liquid chromatography where solvent velocity is controlled by the pumps. Um, yeah, that's should not go too further about that. Then we will discuss about this gel electrophoresis in more details in the next slides. But a little bit of yeah, just to you that you run your samples with the plus and minus with the flowing from bottom to from top to bottom, from negative to positive, and this is your like mixture of macromolecules, and your macromolecules after the electrophoresis is done from the gel, they are separated according to their size. That's the main principle behind this. We will discuss about them in details later. So this is the STS coated proteins. So they are placed on the gels. You can see with the partial separated proteins on it. Direction of migration is happen. Stains to visualize your separated bands, and each protein is separated according to their different size. Smaller size proteins at the bottom and larger size at the up. And the SDS molecular structure looks like this. So SDS page is a key feature, that is the SDS coated have the same electrophoretic mobility, thus molecular sieving effect is major parameter that causes separation of parameters. So at the end your SDS page has different protein structures, like I show you CTGF on that day. I was explaining that 20 minutes talk to you about how different proteins showing different structure. With the help of SDS page you can see these different protein level of expression in your samples. On the side these are the molecular marker ladder, so each size tell you like how much kilodalton of your sample is present. Okay, then you can keep these datas for future also by clipping them, preserving them in your polythenes, or like clipping like that for your samples for the future. So this is the fluorogram of 14C carbon label cellular protein crude cell extract. So you can see some results coming out of it. And this is for classic Comosite dye stain formulation. This is Pierce gold blue stain segments, and this is also Pierce gel gold blue stain reagents. These are stained for 1 hour, and this is then de-staining for 1 hour. No bands were absorbed prior to de-staining here. Bands were clearly visible in the staining tray in this case. And after the wash enhancement step, crystal clear background extensively to 8 nanogram was present in this case. Then comes the isoelectric focusing gel electrophoresis. So this is based on the pH actually, how much is the pH of sample is. Based on that, your samples will be separated. So you have high pH samples, lower pH samples. At one point they will be at the isoelectric focus, they will be neutral pH. According to that pH they will settle down there, and from there we can later remove them with the help of 2D gel electrophoresis. So these are the results of the SDS page, and these are the results from the isoelectric focusing gel electrophoresis. So here we can see a better resolution of your proteins as compared to the SDS page. So they are far better technique of the as compared to your SDS page. So before we start about to talk about 2D gel, we have to precipitate our proteins. That is quite important. So now we are talking one of the technique how to precipitate your protein for 2D gel electrophoresis. We will have a one small protocol. I think there is no sound in this. So we have a extract pro protein isolation kit. So we add this 1 milliliter extract pro to cell samples. So they have some reagents in it. They don't tell us to because it's made by the company. They just keep it secret from us. Secret recipe by the Visual Protein Company. So we vortex for 15 minutes. Then centrifuge at 12,000 G at 4 degrees Celsius at 10 minutes. So inside temperature is 4 degrees Celsius. So that's advanced the centrifuges are actually. Then transfer the supernatant to the new tube. Take 200 microliter of chloroform and mix with the supernatant. Shake it, shake it, shake it for 15 seconds. Then incubate it at room temperature for 3 minutes. Then again centrifuge 12,000 G 4 degree for 15 minutes. there will be a lot of centrifugation. So you have aqueous phase, organic phase, and interphase. So remove the aqueous phase carefully from the top. Then add 300 microliter of ethanol to your interface sample. Then shake it for 15 seconds. Incubate, then centrifugation at 2000 G 4 degrees Celsius for 5 minutes. Now at lower G. So divide equally the supernatant to the new two tubes. So add it to the two tubes the supernatant. Now comes the step of protein precipitation for 2D gel electrode. Add isopropanol 750 microliter in each tube. Close it. Shake it for 10 minutes and then incubate at room temperature for 10 minutes. And again centrifuge for 16,000 G for 4 minutes. Then discard the supernatant carefully. Discard it all. Then add acetone to it, 1 ml of ice cold acetone, and you can see your pellet, your protein being precipitated there. Then vortex the pellet for 1 minute. So bacha, this is proper protocol. Whenever you do any experiments, you have to go through each step like this. If you make any mistake here and there, your whole experiment will be kaput. Yeah, it's all destroyed. So take care of that, please, when you're doing experiment, and discard the acetone carefully. Then let it dry for some time in the air, I guess. Okay, we repeat this acetone wash twice. Then we do open it for some time, and then you have your pure precipitate of your protein in the Eppendorf tubes, which could be used for 2D gel electrophoresis. Okay. So now we run our isoelectric focusing gel there. In previous sample we have shown you. So from pH 4 to pH 10, and then we change the shape of it. Then apply on the top of the SDS page gel. And then it run for the SDS electrophoresis. That's why it's called as 2D electrophoresis. Uh, because first we run our separation by first dimension, then we're gonna separate the protein by second dimension. So the here side is the higher pH, these are the lower side pH samples, and they got separated with the STS page like this. And this is how your samples looks like. With the isoelectric focusing and STS page together, these are the protein dots that are showing your protein expressions of your whole cell extract. All the proteins will come out there. Each spot here is representing a protein. So this was done by Farrell in 1975. High resolution two-dimensional electrophoresis of proteins. It was discovered in Journal of Biological Chemistry. Uh, so now last slide. Here we will be seeing how to do this 2D gel from start to finish. It's a long one. So Enjoy this video.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply sample and rehydrate the IPG strip. The second segment will be on how to equilibrate the IPG strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality two-dimension gels. After sample preparation, the first step to 2D is to rehydrate the IPG strip. And I'm going to demonstrate this process using Biorad's 2D starter kit. The 2D starter kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, CHAPS, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 IPG strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all IPG strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG strip into the focusing tray. An IPG strip has 2 sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip on the negative side. You'll see the other side has a positive side. The negative side is opposite of that. And you can always tell when the IPG strip is facing down, but you'll be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before the step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the IPG strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode of the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first, remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C. The steps following rehydration and focusing of the IPG strip are equilibration of the IPG strip in preparation for the second dimension gel, which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the IPG strip to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the IPG strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the IPG strip, turn it over so the gel side is up, and place the IPG strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the IPG strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the IPG strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the IPG strip. Place the IPG strip into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1 to each lane of the rehydration tray that you have a strip. Place the rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Bio-Rad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, Place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. And do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the die front has reached the bottom of the gel. We're now going to remove the gel from the Criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I open the gel cassette, I use forceps to remove the IPG strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kamasis and silver stains, to the fluorescent stains, flamingo and cypro. In this case, I am staining this gel with BioSafe Kamasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kumasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kumasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kumasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved.
Thank you very much, sir, for presenting such detailed, well explained experiments. Um, So this is how it is done in real lab also. Whenever you're doing a Western blot, SGS page, electrophoresis, or any chromatography technique, our sir staff scientist from BioRed has explained it in really, really massively good manner. I have to admit. So whosoever are still with me, I hope you had a good learning from all these workshops. I think you have understood the basic principle also afterwards, that with the first you run on the isoelectric focusing your with the help of this pH based 1D gel, then you run the 2D gel on the gel, then you see your proteins like coming on this case. So this is all the proteins that are present in your sample, you can see in there. So this is a summary of homogeneous, salt fractionation, ion exchange chromatography, molecular exclusion chromatography, affinity chromatography. This is how see bands actually. This is the comparison of one sample into the different way of results that we see. So this is a single band on SDS page can be accepted as a criterion for pure protein. So this is pure protein. In affinity chromatography, we got a very pure protein about our from our experiments. So, and these are the various total how much the data comparison between all the chromatographies, uh, and the total activity, specific activity, yield, and purification level. And this is last not the least, is your technique. We still have 5 minutes. I want to finish with MALDI-TOF today. This is done to check a laser beam. In this case has been protein embedded in the matrix, ionized proteins accelerated towards the detector. So like this, protein is sample is ionized. Your samples are put in the probes like this. This is your matrix. Your samples are being hit by the laser beam like this. Then there's an ion source. Then flight of tube is there with a positive charge. Lightest ions arrive at the detector. Then detector will read with the help of transient recorder. Laser will trigger a clock, and then with the laser we will see the results on like this. So this is how your whole things works like. And then at the end you see your results in the form of again some peaks are there. Each peak belongs to some protein. So like if you see 5C73573.9, that belongs to insulin. If you see a peak of 18364, then it belongs to the beta lactoglobulin. This is how you identify different proteins. So
Worker MALDI-TOF process. MALDI-TOF analysis provides additional structural information for target molecules. As MALDI-TOF, MALDI-TOF is a two-phase process. Fragmentation can occur by application of higher laser energies, known as LID, laser-induced dissociation, or a background gas can be introduced to promote ion collisions. This process is known as collision-induced dissociation, or CID. Samples are co-crystallized with a suitable matrix on a sample carrier. The samples are vaporized by the laser into a vacuum while being ionized at the same time. During this process, the sample molecules can become excited. The now charged and excited particles are accelerated through the application of a high voltage, and fragmentation can occur as they travel through the flight tube. In this process, precursor ions and their respective fragments travel together at the same speed. Based on the expected time of flight of the precursor ions, specific ion packages may be individually selected, and all other ions blocked from continuing through the flight tube. The selected ions are then re-accelerated, with the precursor and fragment ions traveling at different speeds towards the reflector detector. The ions travel via the reflector towards the detector, and thus a TOF spectrum is recorded. The detection of the selected precursor ion and its fragments creates a TOF spectrum. Whether fragmentation occurs through the use of higher laser energies or a collision gas, the structural details derived from the MALDI-TOF spectrum provide a deeper analytical insight of the targeted compound.
So that's it for today, I guess. We could not, sorry, manage to make Cas9 gel electrophoresis and PyMol thing. We will continue them tomorrow. Sometimes my streaming could stop suddenly like yesterday, so I don't know why it happens. So I will share all these slides on Telegram. I think I can share it now itself so that we do not forget later. If let me know if videos are working. I will also share the videos later. But it will be a lot of data for all of you. But anyhow, Telegram can handle it. So you learn all about protein today. I hope you have enjoyed today's class. The main reason, oh, it's a big file, 352 MB. So can I share mainly on the MBBT series only, or do I have to share things on the other one, on the internship program? I think you both are equal number of students. So can I share only on the one platform, because there will be a lot of data? No, but J proteins don't denature in that case, because it's being prepared in a solution so that it could be protected. So I will share only on the MBBT series 1. That's it. Everything I will share on this. Great, great. Not 2 times. Internship group also. Do I have to share in the internship group also? Because data is a lot, so it will take a lot of whole night for me if I share it twice. Okay, I will share in both then. No worries. Let it be.